Kia ora koutou, welcome. Thank you for joining us in this session of the Thriving Cities webinar series. My name is Rachel Nepia and I'm the Program Manager for People, Cities and Nature, and I'll be your host for today. This series explores what it means for cities in Aotearoa New Zealand to support truly thriving biodiversity and resilient urban ecosystems. In today's session, we're going to explore urban forest soil ecosystem restoration with two speakers from the University of Waikato. First, we'll hear from Dr. Kiri Wallace. Kiri is a restoration ecologist at the University of Waikato and a director at EcoIndex. She works at the interface of ecological science, business and government to foster biodiversity investment across sectors. After Kiri, we will hear from Dr. Andrew Barnes. Andrew investigates how habitat degradation and restoration impact soil food webs. His research highlights the importance of protecting and restoring soil biodiversity for ecosystem and human health. After both of those presentations, we'll have some time for questions, so please use the Q&A function at any point in the webinar to send your questions through. We'll get to as many as we can at the end. So first up, I'll turn the time to Kiri.
Great, thank you so much, Rachel. It's my pleasure to kick this off. Kia ora koutou katoa. So this is our presentation today titled Urban Forest Soil Biodiversity and Restoration. And I'm going to kind of introduce us to some of the basic topics, and then Andrew is going to dig a little deeper with some case studies showing some of our research at People, Cities and Nature. All right, next slide, please. Okay, so just first, some special thanks, of course, to our host institution, the University of Waikato, our funding institution, and our research team, which is led by Martin Breed. Next slide. 4 things are going to be what we cover today. So forest soil biodiversity types, who's down there, basically. Why do we care? How can we find out what's there with some monitoring? And we'll unpack that with some case studies. And then if we want to restore soil biodiversity intentionally, what can we do? Next slide, please. Okay, so first of all, just a little bit of an update, if you didn't already know, that about 60% of Earth's biodiversity lives in the soil. So this is pretty incredible, but we don't actually know about a lot of it, and it's a really rapidly expanding area of research. It's kind of important, though, of course, because it's right under our feet and it affects everything above ground, and you'll kind of see why by the end of the webinar. So pictured here are kind of the main groups that we'll talk about today and that are out there. The invertebrates, probably most of us are fairly familiar with. We see them because they often will come above ground or they live in the very top layer of the soil. So probably a lot of us have an early childhood memory with an earthworm, right? So that's where it starts. But in the rest of these black boxes, you can see a lot of the incredible life forms that we don't get to see, at least not with the naked eye, or not very often. So for fungi, for example, of course, we see the mushrooms, fruiting bodies from time to time if we're in the right season of the year and we're in the right place outside. But we don't see the hyphae, the white bit in the image here that lives in the soil, the main part of that organism that does a lot underground. And of course, the bacteria, the protists, the archaea, and the viruses, we need a microscope to be able to see some of this and other techniques that we'll unpack to learn what they're doing and who's down there, who they are. Next slide, please. Okay, but first a little bit about why do we care about these mostly invisible things below ground? Well, one first reason is just the international agreements that we've signed up to as a country and the amazing initiatives that are happening in this space internationally, as we all realize how important soil biodiversity actually is. So Aotearoa is a signatory to the Kunming-Montreal Global Biodiversity Framework. Always a mouthful, say that one. And part of that is about soil biodiversity. So here you can see a screenshot of this lovely handful of soil and worms and other things that we can't see in there. And this was released in July of this year, an announcement about the Global Soil Biodiversity Observatory, which is an initiative of the Kunming-Montreal GBIF work. So a lot of other countries are getting in on this sort of work. They see the importance of it, and I think we should probably too. Next slide, please. Okay, but why else? What are the amazing above ground effects that all of this life actually has that we may not realize? A lot of us here may really like plants. I do. It's pretty much my main area of expertise. I also now really love soil biodiversity because I realize how important it is for plants. So I'll just highlight a few things here. You could have a webinar on each of these because there's just actually so much that we're learning. Invertebrates, for example, they are fantastic at aerating and mixing up the soil. This is bioturbation. This helps nutrients move around and helps cycling happen. That's actually really critical, and soils don't do well without that. Fungi, we need these guys to break down tough substances like lignin, so wood, basically, and unlock nutrients in that wood, dead trees, for other plants to grow. For bacteria, one of the more famous examples is the nitrogen fixation that they provide. So of course, legumes, we think of like kowhai trees. They have those lovely little root nodules with those symbiotic bacteria. And that's just the tip of the iceberg for what bacteria can do for nutrient cycling. And then finally, the protists, virus, and archaea get smooshed together, but they're actually all really important in their own right. And did you know that archaea can promote methane and nitrogen cycling and carbon fixation? So pretty important things that we hear about in other contexts, climate change, for example. And here we have these creatures right below our feet doing this kind of work for us. So yeah, next slide, please. Now, because of my plant background, I usually think about restoration plantings of urban forest in kind of a successional theory framework. And we're all probably familiar with this. Starting on the left side of the green line, we do a planting, or even there's a disturbance and then natural regeneration happens. And then that forest grows through an early succession, mid, late succession kind of timeline. Or at least we hope it does, right? We want to see it get to this stage where there's all these beautiful, huge trees and all the animals and the whole ecosystem that can be there. And we find that the soil ecosystem, kind of below the green line, can follow a similar trajectory because these two things are so intricately linked. So degraded soil may be the scenario at first when we do a planting. It could be compacted, lots of extra nutrients that aren't needed, not much organic matter, not much living there. It's kind of a bit of a mess, bit of a dead zone maybe in a lot of ways. And then we see soils go through this stage where there can be early recovery, where there's more of that aeration and structure of the soil forming again, more organic matter inputs like leaves from plants or bark from plants as they grow, or even sloughing off of root tissue. And then we get into stages of advanced recovery where ecosystem functioning is starting to really pump along. We've got interesting specialist species coming back to colonize the soil, and they're using those new niches that are being formed. And of course, eventually we hope for that mature soil ecosystem, really biodiverse, really flourishing life, lots of productivity. So all the sunlight's being captured by the plants above ground, gets pumped down into the underground space and is fueling that to really be working well. And of course, it supports above ground forest maintenance. So essentially, good soil biodiversity is going to help our plants survive and grow and ultimately reach that forest ecosystem state that I think we all really want to see. Next slide, please. All right, so now hopefully you're convinced this is great and you want to learn what you've got in your various patches out there. I've broken this into invertebrates in one side and microbes in another because they're both kind of different approaches, really. But for both of them, the green box here is where I want to draw your attention. It's really critical to remember you need your end question in mind, and then choose your taxa, choose your measurement method accordingly, and realize that it's kind of important to think about sampling analysis and your interpretation and application at the end. Just sampling and a plan for sampling by itself often ends up being can be a waste of time and money, to be honest, because nobody really understands what you sampled for or what it means. So bringing it full circle is really critical. For invertebrates, you can measure really every few years is good to see sort of any trends, any changes. And there's a bunch of different ways pictured here, from pitfall traps, where on the left, where literally there's a hole and things fall in the hole, and then you check what's in there. There's litter sieving, which is a little bit more complex, figuring out what's in that top litter layer and maybe a little bit of the soil at the top there. And you can core. There's a photo there, the guy with the white gloves, and that's getting down more into the layers of soil to really see who's down a little bit further. You can put that into a Berlese funnel with a heat source, and that draws those invertebrates out. Yeah, thanks for the mouse moving there, Andrew. So also eDNA is being used to figure out who's in the soil, but it has some major limitations that we will unpack a little bit later. And finally, there are some resources out there if you're interested in getting into like what kind of invertebrates might be in my soil. So there's this link here, the Pocket Guide to Soil Invertebrates, and I think we'll be sending this around afterwards so you can access that, or maybe we can get the link in the chat. Next slide, please. Okay, now the microbes. Microbes, of course, are a little bit tricky. You can see from the photo of the person holding the soil going, what? I don't see any, I don't see them. You can't, right? So we have to use different approaches for understanding who's there in that handful of soil. And guaranteed, there will be a lot there. So these can be measured a little bit more often just because the populations are really dynamic. They reproduce quickly, they change quickly. But they're harder to sample and to analyze and then to interpret the data set you get out of it. So if you're kind of just wanting to dip a toe into the water of the microbe scene, you could just take fungi to start with, go for a walk, observe the fruiting bodies, look for what mushrooms are there and what functions might they be doing. So you find a mushroom, identify it, it's a decomposer or it's a symbiont with a tree, it's helping it grow. This, of course, is a survey of really limited use, but it just helps you get used to thinking about what's down there, what it's doing. Then there's PLFAs for fungi and bacteria, and these are more about the biomass of these organisms underground, not so much who's there, but still one more kind of tool to have in the toolbox. And then, of course, for the rest of them, we really need to get into the molecular genetic techniques like eDNA, metagenomics, metatranscriptomics. Next slide, please. So these techniques, I won't dive into them here too much, but they're really growing quickly. The variety of them, the ways in which we can do them. You can see here that the data volume and the processing needed also has increased like exponentially. So it's a complex and exciting area. I think it's just going through a teething stage in a lot of ways, and there's a lot of exciting things coming next here that may help us make this more accessible for the everyday person. But I'll leave it here and hand over to Andrew, who will dig deeper into some of our research case studies. Over to you, Andrew.
Thanks, Kiri. Kia ora tātou. So I guess I'll just take over, like Kiri said, to sort of talk about some case studies. We'll try and just do that in a few minutes to give a very quick kind of whirlwind overview of what we learnt about I suppose the succession of soil biodiversity in sites around Aotearoa. So we've been working in a number of sites, sort of from 70 to 80 sites around cities or urban centres in New Zealand, as you can see on the map there, with sort of varying numbers of sites, from kind of 8 to 9 or so, depending on the city. And within each of those sites, students like Bibishan, pictured here, Bibishan Rai, has been working on these sort of 10 by 20 metre plots. And within those, we've been taking kind of multiple samples across those sites to look at what soil invertebrates live there, such as microfauna, like these nematodes pictured on the right, macrofauna, so those are the largest ones, so typically larger than 2 millimeters. So these isopods, as an example, or pill bugs, slaters, whatever you call them, but including spiders, beetles, etc., millipedes, centipedes, but also the vastly diverse mesofauna, which we kind of tend to just know as like mites and springtails, but these are incredibly diverse groups that carry out a range of very different functions in the soil. Look, just to give an overview, essentially what Biebeshan found during his PhD is that while forest age certainly has an effect, so the older the forests get, we see changes in abundances of invertebrates. There's a whole lot of other factors going on, and we can see that here in this figure. So all the points to the right of this zero line indicate that there's an increase in abundance, and to the left indicate that there's a decrease in abundance with these different sort of predictors. So we see forest age, the older the forests get, we see sort of trending increases in predators and omnivores, and also the overall community of all invertebrates in the soil. But interestingly, we see decreases as well. So detritivores, for example, reducing in abundance in those older sites. But this is really context dependent. So huge differences depending on soil temperature, canopy openness, seedling abundance on the forest floor, the mean size of trees, how many tree species there are on a site, and just the total sort of above ground biomass of trees as well. So it's a very complex picture, and it's not as simple as just plant it and they will come. Where the older it gets, the more we'll see. And so sort of follow on from that, looking at Grace Mitchell's work, who did her master's, looking at the microbial part of the soil food web, or the soil community. What we can see on the x-axis of these figures are just the number of years since these sites were planted. So the further we get to the right, the older they are. And overall, we see quite large changes in the composition of bacteria, archaea, fungi, and protists, so those sort of main groups that Kiri talked about earlier. And just noting these are hyper-diverse groups of organisms. So for those that aren't that familiar with microbes, within each of these we have these the phyla, and those phyla are changing in the relative representation and their sort of relative abundance, so to speak. And so this is from metabarcoding data, which also might be known as eDNA. Essentially, it's barcoding information from DNA that's found in the soil. And I guess one question that comes out of this work is, what do we do with this information? So we know things are changing. What does that tell us about, are these soils improving? Are they getting worse? Or are they not doing anything at all? And I suppose just to add to the complexity of this overall picture, that we're looking at highly complex networks of species that are interacting with each other. And so another thing that Biebeshan found from his PhD is that this, as sites get older, the influence of the age of that restoration planting is quite complex. And so it's affecting organisms at different trophic levels. So whether they're decomposer organisms, omnivores, so they feed across different sort of potential resources, or predators, they're affected in different ways by the age of the site. And that is through these sort of mediating effects of the environment. So for example, how large the trees are on the site, how many tree species there are, the depth of the litter, and the soil temperature. And there's also this sort of simultaneous effect of the microbial community on the invertebrate system as well. So ultimately, this is just to show you that soil systems are extremely complex, and there's this sort of network of interacting functional groups that we need to kind of consider when we're thinking about how these systems are changing and what they're doing. And so, if we distilled just a few kind of conclusions from all of that work, what should we really be aiming for in terms of, when we're restoring a site? Pretty much three simple things. Larger trees are better, so we should be thinking about planting trees that will eventually become large. We want as many species as we can get. That's sort of reasonable in terms of the sort of typical assemblage of species we might expect in a forest for that region. And we really want to be looking at more leaf litter, so basically more dead organic matter on the forest floor. And Interestingly, Grace also then looked at some functions associated with the microbial part of the community. So she looked at microbial respiration, essentially just the total activity of microbes in the soil, and found that there is this sort of trending increase. So the older sites get, the more activity there is in the microbial part of the soil food web. But even more interesting, I think, she found that the older the sites are, the more variation there is across that 10 by 20 meter plot. So if you go from one side of the plot to the other, there's really large variability in the respiration of the microbial community in those older sites. So older sites mean more spatially variable forests. And a current PhD student, Stella Brachman, is finding that variability is really important for ecosystem resilience. So she's measuring this thing called functional insurance. Essentially, what this means, so high functional insurance indicates that if we lose some species, we can still expect the functions those species carry out will continue to be provided in that system because there's other species that kind of come in and fill that role. And so this metric of spatial variability is showing that it's quite important. So older sites, they're more spatially variable and they're quite likely more functionally resilient if there's some other disturbances that come along. So I just want to sort of finish up here by pointing out that obviously, if we're to keep track of and monitor soil biodiversity, that's going to be a very complex, potentially difficult thing to do without the right sort of expertise and equipment. But there are some sort of more rapid techniques that we can measure the functions carried out by these groups, such as bait laminar strips that measure feeding activity of invertebrate decomposers. This popsicle stick assay, so you can just buy popsicle sticks at your local store. And if you bury these in the ground, you can measure the weight before and after you bury it and calculate mass loss of wood, of dead wood in a system. And many of you might be familiar with the tea bag index, where you bury tea bags on the ground, so green tea and rooibos tea, and look at the mass loss of that tea. And so that sort of partials out microbial decomposition. And it can also indicate, for example, carbon stabilization, which is really important for carbon sequestration. So ultimately, the aim here is really to determine the feeding rates of soil organisms, and it tells us something about the different sort of major functional groups in the soil without necessarily having to identify all of these organisms, which is, again, challenging to do and requires some very specific expertise. So just to wrap up here, I just want to remind everyone, of course, that the soil community is going to keep changing over time. And so when we restore, if you have the capacity to look into soil biodiversity and see whether or not you're having an impact below ground, it's important to keep measuring over time, even if it's every few years, just to kind of keep a finger on the pulse. Are you actually seeing changes that we expect to see below ground as well? And so, yes, it's a very, very rapidly growing space, but just some key things to consider. So firstly, how to restore soil biodiversity. So like I said earlier, plant diversity is really key. Habitat variation is critical for below ground biodiversity. And the other thing is allowing buildup of dead organic matter, particularly like dead wood and leaf litter, and potentially even adding this kind of thing like dead wood into early stage restoration sites. Messy is good, right? Soil organisms need habitat heterogeneity for that functional resilience I talked about. And encouraging establishment of these large, long-lived trees. So really forward planning and thinking about what trees you might be planting that one day will grow into large sort of late successional species that have quite a strong impact below ground. And so I'll kind of leave the last bit, I suppose, to questions if there are any about it, but I just want to point out that we should also be considering how we monitor below ground biodiversity, and it's really critical that we're looking at the abundance of biomass of below ground organisms rather than just species detection, because species detection alone can't really tell us about the relative role that these organisms are playing below ground. And we just know so little about below ground biodiversity. We'll be getting lots of sort of undescribed names on those barcodes that don't really tell us what's below ground. Ultimately, if we can't do that, monitoring functions are really essential. Okay, so I'll leave it there. Ngā mihi nui, and look forward to any questions.
Awesome. Thank you so much, Kiri and Andrew, for sharing your work with us. We will open the floor to questions, so please put them in the Q&A chat. Our first question here is from Kate Lee. It says, do you see more variation in biology between urban areas or between types of sites? For example, like, are you seeing more variation between cities or between parks and veggie gardens, things like that?
I can take that one if you like. I think that's a hard question for us to answer from this work that we've done within the People Cities Nature Project, because we've specifically been looking at sites that are being restored with native plants to eventually sort of, I guess, take that trajectory toward a forest-like ecosystem. But there is a fair bit of work looking at kind of variability in that biology among ecosystem types within urban centres, and actually cities have surprisingly heterogeneous soil communities because environments can be so extreme. And so, yeah, certainly, there is research out there showing that if we look, say, for example, between like park-like green spaces versus, say, playgrounds or gardens, there is a huge amount of variability, and probably much more than what we would see like among those different successional stages of these planted forests. I don't know, Kiri, if you've got anything else you want to add to that.
That was great. I just had one thing to add that I've learned in the last few years during this research that I didn't realize, and some of you may not, that a lot of the microbes in the soil can become aerosolized in the air, and they fly all around. They float all around on the wind and stuff and get dispersed quite readily. So they're by no means like static or stuck down there underground. The worms probably are.
Thank you. Our next question is kind of about soil preparation. So if you're looking to create a dense, multi-layered native forest in a new reserve or on former farmland, are there any soil tips beyond the usual topsoil requirements that could help to create a biodiverse soil and a thriving planting?
We talked about this a bit. You go ahead, Andrew. You were thinking on it more than me.
Sure. Yeah, I mean, I think like you'll find lots of tips out there. I guess it's always kind of partly ecosystem dependent, but generally speaking, I think often like with soil, we're looking at sort of getting species in that do so-called niche construction. So planting plants, I think, is a really important prerequisite. Just getting plants into the ground, planting them densely, and making sure that there's a reasonable diversity of plants as well. So as opposed to, say, planting just pure like manuka across your whole site, really think about a good mix of early successional species that you can plant to ensure that diversity from the early on. And probably just simply putting down like a good layer of mulch so that we're not just putting plants in the ground with just bare soil everywhere. That is absolutely critical for especially for soil invertebrates, is that additional habitat layer. And soil invertebrates are also known to be quite important for facilitating microbial activity as well. And so really you're trying to get the whole system restored. And a layer of sort of dead organic matter, dead wood, like mulch, is going to be really important to get things going.
Awesome. Thank you, Andrew. That's about all the time that we have for questions now. We have a lot more questions in the chat, which I will talk to our researchers about getting some answers for to circulate to the group. So thank you so much for submitting those questions. And thank you, Kiri and Andrew, for sharing your kōrero with us. And thank you to everyone that joined today. We really hope that you're leaving the session with some new insights into just how important soil biodiversity is for our urban restoration. Our next talk in this series will be in December about the role of maramataka in urban restoration, and that will be with Dr. Erina Walker and Wiremu Puke from Ngāti Wairere. So until next time, kia pai tō rā.